Hello, is it audible? Host, sir. Hello, hello, hello. Seats are on the line now. Hello, hello. BRAOU host. Is it audible? Hello, hello, hello. Jyoti. Hello, Jyoti Kalyan. Hello, Jyoti Kalyan.
Yes, sir.
Is it audible?
Yes, sir.
Right. Yesterday, last class we completed about what you completed last class. Jyoti, did you attend last class? Hello, hello, Jyoti. Hello, did you attend last class? Right. Last class we completed about DNA structure, nucleus and DNA structure, Watson and Crick model, DNA Watson and Crick model, and also we started DNA replication also last class. DNA replication, right? What is DNA replication?
Making of other copies.
Right. Are you able to see the slide?
Yes, sir.
Is it
It is cell
visible
wall
to you?
structure. Yes, sir.
No, no. DNA replication slide visible to you?
No, no, sir. DNA replication slide is not there. Cell wall ultrastructure is there, sir.
Now,
No, sir.
now,
Yes, sir.
right. So as you said, what is DNA replication? Replica means the same copy. That means one DNA strand prepare another DNA, right? That is DNA replication. Let's see the process. What are all enzymes involved in this? First, let me complete one by one enzymes. After that, we completed the process. So the first, let's discuss about replication enzymes. So there are so many enzymes involved in DNA replication. That means the making copy of new DNA strand from old. The first enzyme is DNA helicase enzyme, DNA gyrase enzyme. Are you able to see this enzyme slide?
Yes, sir.
Right. Note down all these enzymes. First of all, note down the first definition. DNA replication definition. One DNA strand makes another DNA. This process called as DNA replication. So in DNA replication, mainly some enzymes involved to carry out the replication process. First of all, DNA helicase and DNA gyrase. SSB means single stranded binding protein. Single stranded binding protein. Right. First, DNA helicase. First, what is the nature of DNA? Already we discussed in DNA structure. Always DNA coiled nature, supercoiled nature, or uncoiled nature. Winding or unwinding. How is DNA? According to Watson and Crick model, how is DNA? Always two strands winding or unwinding nature.
Two strands, sir.
Two strands winding, wrapped
Yes.
around each other, right?
Yes, sir.
They are wrapped around each other. That is called Watson and Crick. Here also you discuss about degrees also. Here the 360 degrees turns around each other. So first of all, before synthesis, before making one DNA, we should remove the supercoiled nature, right? We should remove the supercoiled nature, and also we should remove the how attached two stands. Hello? How these two stand? Already one stand 5 and 2 3 and 3 and 2 5 and already told you both stands anti-parallel direction, right? Last
Yes,
class we
sir.
discussed both stand anti-parallel direction and each stands attached with hydrogen bonds and they
Yes,
are
sir.
super coiled. So that's why to prepare new stand here it should remove the hydrogen bond and it should remove the helix also, right? First of all, for example, this is one stand, this is another stand. Always, for example, this is 5 and 3 and 5 and 3 and 3 and 5 and. So here attached with hydrogen bonds, right? Hydrogen bonds. And actually the DNA stand always coiling nature, isn't it? Right? Always coiling nature. Right. This is the DNA. So what is replication? One DNA strand prepare another DNA. That's why first of all, unwind. This is winding nature. Winding nature. Unwind DNA. Unwind DNA. That means the separates the two parental strands and establish replication fork. Establish replication fork. For example, this is one turn. For example, if it is remove, here the formed as a replication fork like this. One turn remove. Rest of the same. So this is called replication fork. So first of all, with help of the LKase enzyme, these hydrogen bonds removed in between two strands removed, and also as a result, this folding nature unfold, this winding nature unwind, this winding nature unwind. Now it's formed as a replication fork. Now the next enzyme DNA gyrase, are called as topoisomerase. Topoisomerase. For this Alkase reaction, it required one ATP. For one ATP required for the hydrolysis of hydrogen bonds. That means removal of hydrogen bonds and also unwinding the two strands. DNA garages topoisomerase, the same like a Alkase enzyme action. It is also supercoiled into duplex DNA head at the place of the replication fork. Now the function of DNA gyrase relax the supercoil. Now, for example, here the replication fork formed. What happened here? The behind the replication fork, another folding, another winding is present. Right? The DNA gyrase enzyme, the relax, that means somewhat loose. Actually, these wrapped tightly around each other with help of the gyrase enzyme, the folding nature, it's make loose, the tight nature make loose. That is that called as relax the supercoil. So this function is done by DNA gyrase enzyme. Here the DNA gyrase topoisomerase two types, topoisomerase 1 and topoisomerase 2. So anyway, first helicase enzyme remove hydrogen bonds and remove the winding nature, and it's make the unwinding. After unwinding, just imagine next folding, next turn. This next turn always supercoiled. Due to the supercoil, it is impossible to remove the coiling, remove the twisted DNA. To remove the twisted DNA, it should untwist. Before the untwist, with help of gyrase enzyme, it makes free. The twisted nature got relaxed with help of DNA gyrase enzyme. What will happen? Next, with help of Alkase enzyme, the hydrogen bonds continually removed, as well as the DNA strand separated continually. DNA strand separated continually. And meantime, single stranded binding SSB protein attached to the DNA strand. The function of single stranded DNA preventing rewinding of DNA strand. What is the nature of DNA? Already told you, always these are winding. If you remove, again winding like a dog tail. If you straight, again it's bend. Here also the nature of the DNA, if you remove, again they start to coiling. But with help of single stranded binding protein, what happened here? Do not fuse, do not rewind. Prevent the rewinding of DNA strand. That is single stranded binding protein function. Next, DNA primase. DNA polymerase can only extend the existing polynucleotide, but cannot synthesize the new DNA strand. Here this for actually we will discuss later about DNA polymerase enzyme to activate to functioning of all polymerase enzyme. Here the required DNA primer, sorry, RNA primer. This RNA primer synthesized with help of the DNA primase enzyme. With help of DNA primase enzyme, RNA primer synthesized. With help of the RNA primer, here DNA polymerase enzyme can do their activity, right? So that is the function of DNA primase. So they are all happened in order. First hydrolyzed bonds removed, unwinding DNA, and next fork got relaxed. Next, prevented rewinding. Next, start synthesizing. What start synthesizing? What start synthesizing? Here the start synthesizing of new DNA strand. Start synthesizing of new DNA strand. To synthesizing DNA strand, to synthesize, for example, this is one end, two strands after separated. For example, this is got separated with alpha of this DNA helicase, DNA gyrase, SSB, 5, 3. Always anti-parallel, already told you. 3, right? Are you listen? Hello?
Yes, sir.
Right. If you have any doubt, ask me. Right, anti-parallel. 5 and 3 and 3 and 5, right? Here always the primer. What I discussed so far, the primer, RNA primer, the RNA primer synthesized from the DNA primase enzyme, right? Right. So the RNA primers attached here, at 3 hydroxyl end are able to see slide?
Yes,
Hello?
sir.
Right.
Replication enzymes. Oh.
Replication enzymes, right. Where is marker? Wait, 1 minute. Right. So there three hydroxy end, 3 OH group, one RNA primer. Here also one RNA primers attached at 3, 3 end. So just follow the marker. Here 3 end, okay. Here primer attached. Here primer attached. After primer attached, now hello, hello,
Yes, sir.
right. After primer attached at 3 end, now recruit the DNA polymerase. Now DNA polymerase 1, mainly DNA polymerase 1. After attached DNA polymerase 1, let's see the function of DNA polymerase. Where the DNA polymerase 1 attached here at 5 end or 3 end? Hello? Where the primer present, there DNA polymerase 1 attached. Where is primer attached with 3 OH group or 5 OH group? Remember always the primer attached at 3 OH. Here also 3 OH, right? Hello?
Okay, sir.
Right, right. Be silent. Please be silent. Make it mute. Thank you. Later, as a result of attached DNA polymerase 1, what happened next? We'll see later. These are DNA polymerases, but in different in prokaryote, eukaryote. Prokaryote different and eukaryote different. First, let's discuss about prokaryote replication enzyme. In prokaryote replication enzyme, 3 type: DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. Here, the first DNA polymerase 1 helps in DNA at 5 and to 3 end direction. At exonuclease activity 3 and to 5 end direction. Simple, remember, polymerization or synthesizing 5 and to 3 end. That is synthesizing 5 and to 3 end, synthesizing new strand. Remember one word carefully: the newly synthesized DNA always 5 and to 3 end. Newly synthesizing DNA always 5 and to 3 end. Next, The same enzyme done exonuclease activity. Exonuclease activity means reverse to the synthesizing. That means in exonuclease activity, removing hydrogen bonds towards 3 and to 5 end. Here, 3 and to 5 end, removing the hydrogen bonds, but the synthesizing of the DNA strand anti-parallel direction 5 and to 3 end. So the two functions done by one. One is synthesizing polymerization of DNA. Second, exonuclease activity of DNA. Next, DNA polymerase 2. This is not involved in replication, but it can repair mechanism. I will tell you how the repair. Repair means if any gaps occurred in DNA strand that can repaired by DNA polymerase 2. And 3, this participate in DNA replication and show synthesis in 5 to 10. This is also involved in DNA replication. 3 also like a 1, but only one direction. The direction means only one direction, 5 and to 3 and direction polymerization reaction only, but not involved in exonuclease activity like DNA polymerase 1. So these are the prokaryote replication enzyme. And let's see the eukaryote replication enzymes. In eukaryote, five types of DNA polymerases are present. They are alpha polymerase, beta polymerase, gamma polymerase, delta polymerase, epsilon. First, alpha polymerase helps in replication of DNA strand. Beta polymerase helps in repair mechanism. Gamma polymerase helps in replication of mitochondrial DNA, but not nucleus DNA. In mitochondrial DNA and chloroplast DNA, the replication is done by gamma polymerase. Delta polymerase involved in DNA replication during elongation phase of the DNA. During elongation phase, also in DNA repair. That means strong mistake. So mainly delta polymerase can repair the DNA. Delta polymerase, the function of delta polymerase in DNA replication can repair the DNA. That means fill up the gaps. Epsilon, involved in DNA replication and also DNA repair. Right. Epsilon and delta polymerase both are involved in DNA replication and also DNA repair. So these are the enzymes involved in DNA replication. Right. Now, let's see in diagrammatically. Let's see diagrammatically. Is it visible to you?
Yes, sir.
Right. So, for example, this two stand, right? Here parental stand. Are you follow the marker? Hello?
Yes, sir. Yes, sir.
Right. This is parental stand. This is newly synthesizing stand. Right. Actually, we discussed one by one. That is helicase enzyme first, topoisomerase second, SSB. This blue color SSB. And now let's discuss one by one. What is the function of helicase enzyme? First, before replication, first helicase enzyme attached at the DNA fork. Then what is the function of DNA helicase enzyme? What is the function of helicase enzyme? This enzyme? Hello?
It removed the hydrogen bonds.
Right.
Unwind.
Remove the hydrogen bonds and unwind. As a result, formation as a fork. But rest of the DNA the same nature. That means they always unwinding nature. Now, what is the topoisomerase? What is the function of topoisomerase?
Super cold.
Now next attack. Right. Remove the superkile. Actually, this is the superkile nature DNA. Free the superkile nature. Make it free so that the LKs remove the DNA, sorry, LKs remove the hydrogen bonds between the two strands. That is the function of topoisomerase. Next, single standard binding protein. These blue color dots, single strand binding protein attached. What is the function of single strand binding proteins? These proteins?
And they help in preventing unwinding.
Right, help in preventing unwinding. Because if the SSB absent, what happen? If we remove the strands and again winding nature. So this is interrupt the making the DNA. It can interrupt the making DNA. That's why Due to the help of the single stranded binding protein, it cannot rewind, right? Next, next enzyme already told you primase, DNA primase which synthesize the RNA primer. This is RNA primer. Here the RNA primer, right? RNA primer, right? For example, this is 5 and 2, 3 and this is 3 and 2, 5 and this stand, this parental one, two black color parental one. Hello, are you follow
Yes,
my
sir.
mark? Right? This is parental stand and this is parental stand. Okay. The parental stand one is 3 and this end 5 end. Okay, right? Right. What
Yes, sir.
already told you the RNA primer always attached at 3 end, right?
Yes, sir.
Here 3 end. But this is 5 end. Where is 3 end here? Opposite end. This
Yes,
is
sir.
3 end. This is 3 end. Right? So one primer attached here, another primer attached here. Right? What happened after primer attached? What happened after primer attached? Which
Uh,
enzyme recruit? Which enzyme recruit? DNA polymerases.
alpha
Eh,
polymerases, yes.
right. In prokaryote, DNA polymerases, but in eukaryote, alpha polymerase, beta polymerase enzyme recruit. So DNA polymerase enzyme attached primer and the function of RNA polymerase, sorry, DNA polymerase one enzyme synthesize new strand and also remove the hydrogen bonds. Now that always already told you the newly synthesized DNA always towards 5 and to 3 end direction, right? So
Yes, sir.
this is 3 end, this is 3 end. So here 5 end, 5 end. So it should continually synthesize the new DNA strand without interrupting, without breaking, because here the DNA strand synthesizing initiated. Now it is keep going on, keep going on. Now where the fork is present, there stop. Again the fork unwind, again continued without interrupting, continuing. But what happened in the other hand, that is this end, 5 end to 3 end. In 5 end to 3 end, actually the DNA strand synthesized from the 5 end to 3 end direction. Where is 5 end? This is 5 end here. This 5 end, opposite 3 end. Here 5 end. Already told you the primer start at 3 end direction. But newly synthesized DNA 5 end to 3 end. Now here the 5 end. Arrow mark. Are you follow arrow mark?
Yes,
Hello?
sir.
Here 3, here 5 end. Now 5 end to towards the 3 end. But it breaks, but one fold. And again remove the another fold. For example, here 5 end. Opposite 3 end, right? Here 3 end. After primer, here for example, this is 5 end. It should be, this end should be 3 end, right? Hello? Let's change color. This is 3 end. Blue color primer.
Yes, sir.
Here primer. Now primer attract should polymerase enzyme. Polymerase enzyme should be in change color, the green color. Polymerase. Here also polymerase. Now tell me the direction of synthesizing DNA always.
5 into 3 end.
So for example, here 3 end. So 5 end here. Okay. 5 end to 3 end, this direction. What happened this opposite side? This stand? Where is 5 end?
Uh,
Started from new DNA stand from this side or this side? Where is 3
this
end?
side, sir.
Opposite. This side, here only. Here only. So from here to It should be 5 into 3 end, right?
Yes,
This
sir.
is this is 5 into 3 end, this direction, but this turn direction, this is 5 into 3 end, right? After unwinding, this is folding. Due to the SSB and also leakage enzyme and garage enzyme, this fork removed. And again, this stand keep going on continuously without breaking. Keep going on the entire DNA, parental DNA without breaking. But what happened here? The already break. After removing, the new stand synthesized from the this fold. Okay, until this
Okay.
one. And again another stand synthesized from the this stand until this fold. That is the always break into pieces. This is one fold break, another fold break. So this is called as Okazaki fragments. This is called as where it is given or not Okazaki fragment.
It's there, sir. It's there.
Okay, right. This breaks one, two. This breaks called as Okazaki fragments. And here another one third. If it is say fourth, here the fifth. But what happened here? But this end is always keep going on. This is stand leading stand without break leading stand. But the breaking into pieces that is called a logging stand. Remember, this is leading, always lead
Okay.
continuously leading stand. Breaking pieces, logging stand. This stand is called as Okazaki fragments here. Okazaki fragment.
Yes.
Is it clear? Right.
Yes, sir.
Already told you the repair. This DNA also can repair and with help of ligase enzyme and also with help of DNA polymerase 2 enzyme. DNA polymerase enzyme and also epsilon, gamma. In eukaryote, gamma polymerase enzyme, epsilon polymerase enzyme can repair the DNA. But in prokaryote, DNA polymerase enzyme and ligase enzyme, with ligase enzyme attach the two strands. And if any gaps filled it, this gap removed by the or repaired by the DNA polymerase 2. Like this, the complete DNA strand prepared. So one DNA strand, this DNA strand prepared this one, this DNA strand, this one, right? After completion of the DNA, this to mother, that older and new supercoiled and separated into one cell. And on another side, older, new, coiled and separated into one cell. This is called DNA replication process. Is it clear? Hello?
Yes, sir. It's clear.
Right. Any doubts in this?
No, sir. No, sir.
Right.
We can move on, sir.
Right. Right.
We will go for
Next.
next chapter, sir. It is easy types
Yes.
of RNA.
Types of RNA easy. No, no need to explain.
No, sir. One cover leaf model tRNA. This you explained. That's it, sir.
Which one?
Cover leaf model tRNA.
Okay, tRNA. Only tRNA. Right.
Yes, sir.
Okay. So there are three types of RNA. I already told you that is we discussed earlier.
Messenger RNA.
Messenger RNA, ribosomal RNA, and tRNA.
Yeah.
tRNA other name transfer RNA. Right. Right. Are you able to see this picture?
Yes, sir. Yes, sir.
Among the three types of RNA, it is recognized as the smallest RNA. Biggest to largest one, messenger RNA. The smallest one, tRNA. It is contain only 80 to 90 nucleotides. This nucleotides made up of 5 and 2, 3 and all complete nucleotides are just 80 to 90 nucleotides. Out of the total cellular RNA, the portion acquired 10 to 15%. And it is also called as soluble RNA or sRNA or adapter RNA, translator RNA. The molecular weight about 25,000 Dalton. Most widely accepted model of tRNA is clover leaf model. It is look like a clove leaf. That's why it is the model proposed clover leaf proposed by holy scientist. Proposed by holy scientist. This model shows a single stand. Actually, it is appear look like a double stand, but not as double. It is not a two strands. One strand start. This is only one strand. Only one strand. This strand end with three end. End with three end. So which is stands folding? Single stand. This single stand shows the folding nature. It shows the three distinct loops and the fourth one indistinct loop, which is considered as a auxiliary hum, extra hum. Let's say the three loops. One is do loop here, D loop, enzyme recognition site. This is enzyme recognition site in protein synthesis. Opposite T loop. This is ribosome recognition site. Anticodon loop. This is anticodon loop. I will tell you about the function, all enzyme recognition, ribosome recognition, anticodon loop. And here the considered the fourth one, extra one. This is extra loop, right? So this is three loops completed, one extra loop. In the tail portion, three unpaired nucleotide. This is tail portion. This is tail. Actually, except the CCA, rest of the all paired with each other. Paired with each other, but this except this CC and unpaired nucleotide. It present at three end. Presented three end. The CC end is also called amino acid binding site. Here the amino acid during protein synthesis, amino acids binding here. So what is protein synthesis first? Hello?
Yes, sir.
Protein. What is protein synthesis?
Protein synthesis.
What is synthesis means making, protein making, protein, synthesizing the protein. What are the subunits of the proteins?
Uh, Amino acids.
That's it, amino acids. Now, to prepare the protein stand, this tRNA takes the amino acids toward the mRNA stand to prepare the protein. But in tRNA, exactly CC end, exactly three end of CCA attached by amino acids. That's why it is called as a amino acid acceptor site. The loop opposite to the CCA, the loop opposite to the CCA, and shows three. Sorry, this shows unpaired bases, which is constituted. This is also unpaired bases. It's called as anticodon, which constitutes anticodons. There are two lateral loop sites. Lateral loop right angle to the tail part, namely enzyme recognition loop. Already told you, and left side ribosome recognition loop. This enzyme recognize loop as specific site to recognize amino acyl synthetase enzyme. Actually, these are all again repeated in protein synthesis. You don't worry here. Here you just remember the dRNA structure. What is single standard folding? 5 and 3 end. Four loops, mainly three. One extra loop. CC end, anticodon end. That's enough, right? Is it clear?
Yes, sir. It is clear.
Any doubt RNA structure?
No, sir.
Right.
If you draw the structure, that is enough for that, no, sir. Yeah, this
That's
only
enough.
clone
Enough.
or 3D,
Just
3D model.
just only you just write about the this is the smallest synonym soluble RNA, adapter RNA, translator RNA. Who gives the name cloverleaf model? Wholly enough. Next the structure. Loops. Loops name. Loop
Okay, sir.
function. That's enough. That's more enough to because it is a short answer. Just 4 marks. I think 4 or 5 marks. Right, okay?
Yes, sir. Yes, sir.
The next one. Chromosome. Is it clear? Hello, are you able to see the picture?
Yes, sir.
Right, chromosome diagram. So chromosome very important for long question. First, let's discuss introduction about chromosomes. These are the rod-shaped structure. Where are the chromosomes? Where we found the chromosomes?
DNA.
Where is DNA? DNA present inside the chromosome. Where is chromosomes? I'm asking you, where is chromosomes?
Nucleus.
That's it, nucleus. Inside the nucleus, chromosome present. Inside the chromosome, DNA present. Right?
Yes, sir.
Right. So these are the physical basis of heredity. The simple definition, these are the physical basis of heredity, and they are rod-shaped structure present in nucleus. Sutton and Boveri proposed the chromosomal theory, which stated that chromosomes are vehicle of heredity. Scientists Sutton and Boveri proposed chromosome theory. According to this theory, chromosomes are vehicles of heredity. That means heredity transferred to one generation to another generation. Right? The first half-mister identified deeply stained dark red structure in a dividing nucleus of pollen mother cells of Tridus conchia. First observed in Tridus conchia pollen mother cells by half-mister. Next, Waldeyer named them as chromosomes. Who's named as chromosome? Waldeyer. First observed by half-mister, chromosome named by Waldeyer. Right? Is it clear? This
Yes,
is
sir.
about structure. So the structure, size, shape, number of chromosomes different to from one species to another species, both in animals and plants. However, in each species, in the number of chromosomes remain constant, isn't it? In each
Yes, sir.
chromosome, number of chromosome constant. For example, human being 46, Halimsipha 16, chimpanzee 48. So that is the based on the species. So it is remains constant and maintain specificity and stability. Let's see the size, shape, number. Generally, plant chromosomes are bigger in size than animal chromosome. In terms of chromosome size, plant chromosome bigger than animal. Shape, they are different shape. They appeared in banana phase, V shape, L shape, Y shape, J shape. They also you must have learnt in previous classes. Number, already told you constant number. So this is something. The mainly, let's see the structure of the chromosome. Here the chromosome mainly inside the chromosomes, chromanimata or chromatids are present. Chromanimata, chromatids, chromomere, they are all same, but is based on the sector we give the names. For example, actually in this, are you able to see this red color?
Yes, sir. Sir, your voice is not audible.
Just wait.
Okay.
Clear? Are you able to see? Hello?
Yes, sir. Yes, sir.
Just wait. Mark or not. Are you able to see red color?
Yes, sir.
These are called chromatid threads. Actually, they are appeared in they are appeared in where it is different shape, chromomere shape. It's not working. Folding like this, it is called chromomere. Same structure. This is chromomere. If it is long thread like this, chromonema. The same short and thick. This is called chromatid. This is called chromatid. The same structure, but the different stages in cell division, prophase, metaphase, anaphase, it is appear like this. The first in interphase, this the chromosomes, chromatins appear like this. This is called chromomere. This structure called as chromomere. Elongated thread called as chromatids. This is chromatid. But short thread called as chromatid. Short thread called as chromatid. Elongated chromonema. Elongated chromonema. The folding like a necklace. Folding like a necklace. This is called chromomeres. Meres, chromonemata, short thread chromatids. They are the different structures of the chromatids. Chromatids inside the chromosome. This each chromosome, two chromatids like this one and two. Two chromatids. These two chromatids covered by pellicle layer. These two chromatids covered by pellicle layer, right? Covered by pellicle layer. Right? This is one structure. And another one we can see here, telomere and secondary construction, satellite. Here also secondary construction and here primary construction. First, let's discuss from the primary construction. Primary construction or primary centromere. First, primary centromere, the region of the chromosome where the two chromatids are held together. Here the primary construction, two chromosomes, chromatids. Here the two chromatids held, two chromatids held. So this place called as, what is this place? Where the two chromatids are held together, that point called as
Primary construction.
primary construction or centromere. It is a non-stainable part. Non-stainable. If we use the stain, except the primary constitutions, primary or centromere, the remaining part of the chromosome can absorb the stain and appear the stain color. But here at centromere, non-stain region, and uh, the position of the centromere is constant for the particular chromosomes. It is divided chromosome in two arms. This is one. This is one. So at the center point, it divide the chromosome into two arms. Here it is covered by disc-like structure, covered by disc-like structure here. Disc-like structure, protein, disc-like protein, disc-like protein. This is called kinetochore. This is called kinetochore. The function of kinetochore during cell division, here the implantation of the spindle fiber. Implantation of the spindle fiber. That is the function of kinetochore. Right? Next, after primary, secondary construction. Secondary construction. The construction developed other part of the primary. Primary present, other apart from the primary, secondary also present. The chromosome do not bend the region of secondary construction during anaphase. Actually, during anaphase, what happened? Here the chromosome shows bending. Already we can see in diagram. In diagram also chromosome shows bending. But what happened in secondary construction? Is it bending nature or straight? In two strands, in two region, here secondary. Here the bending or straight nature, straight line or bending?
Straight, sir, straight.
Straight. Do not bend. Straight during anaphase. The next one, certain secondary construction are concerned with the formation of nuclei, and they are called nuclear organization. Actually, these secondary construction developed with help of nucleus. Nucleus is the part of the chromosome, sorry, part of the nucleus. From nucleus, secondary construction develop. So this is about secondary construction, secondary centromere. Next, satellite, this one. It is a round knob-like structure we can see in diagram. Round knob-like structure, end of the chromosome, round knob-like body, separate from the rest of the chromosome by secondary construction. You can see the secondary construction also. The chromosome bearing the inside the this region satellite called SAT chromosome or SAT DNA. Inside this SAT DNA or SAT chromosome. Right? Next one. So primary, secondary, satellite completed, kinetochore also completed, a telomere. Telomere, the repulsive end of the chromosome present at the tips of the each arms called the telomere. The function of telomere actually in a nucleus number of chromosome present, there is a chance to easily bind to each other, one to another. But with help of telomere, they prevent the binding of the chromosome one to another. So that is the function of telomere. The polarity end of presence only in eukaryotic chromosome, absent in prokaryotic chromosome. Only the polarity shows, this knob-like polarity shows in eukaryotic, but this is absent in prokaryotic chromosome. Hence, the prokaryotes one end chromosomes fuses with other and form the circular chromosome. If you see the prokaryote the chromosome easily bind each other and it is formed as a circular. But in eukaryote, due to the presence of polarity, telomere do not bind each other. They are individually separated with help of telomere. Right? Is it clear? Chromosome clear?
Yes,
Chromosome
sir.
structure. Anything remain there?
No, sir.
Right. And next one, nucleus. What is time now? Just wait.
5
Right.
minutes are there.
Right. Just wait. Next, nuclear model. Sorry, nucleosome model, solenoid model. Simple. Just a minute. Is there a heterochromatin, euchromatin?
Yes, sir, it's there.
So right. Let's discuss heterochromatin, euchromatin also. Simple. Based on the staining, the chromosomes are chromatin are two types: heterochromatin or euchromatin. Right? What is heterochromatin based on staining? Fully stained or dark stained or light stained?
Okay.
Hello?
Okay, sir.
Tell me. Just see the book. Just see the book. Just one word. Just find out one word. Difference between heterochromatin, euchromatin based on staining, right?
Yes, they are
Dark,
heterochromatin are tightly packed and they are loosely packed.
right? That is one packing. But stained, tightly packed in heterochromatin or euchromatin?
Uh, Darkly stained is heterochromatin and euchromatin is lightly stained.
Lightly, right? That is one. Next one,
Uh,
compactly arranged heterochromatin, loosely euchromatin, right?
yes, sir.
Right. So that is the main heterochromatin and euchromatin. And another important one is nucleosome model or solenoid model.
Folded fiber model is also there.
Folder fiber model, right? folder fiber model. The chromosomes, already told you the nature of chromosomes. Where this diagram? Are you able to see slide?
No, sir.
Yes. Here, nucleosome model. Just wait. Return meeting. Screen share. Right. Now,
Yes, sir, it is visible.
right. So nucleosome model. First, before nucleosome model, we discussed about DNA structure, right?
Yes, sir.
Actually, this DNA wrapped around histone proteins. These are this green colored is called as these pink colored, blue colored called histone proteins. The histone protein DNA together form a nucleosome model. This is nucleosome model. Nucleosome model. Now this nucleosome model closely packed. Closely packed. This is called as solenoid model and finally formed as a true chromosome. This is chromosome structure. Simple. First, let's discuss about nucleosome. This is important. Nucleosome model. This model important. Nucleosome model. DNA and basic histone protein constitute the chromatin. Already told you. DNA and histone. Actually, blue color DNA stand here. Blue color threads DNA stand, and here the light orange color or light brown color. Here, this one. This is histone protein. Are you able to see? Hello?
Yes, sir. But your cursor is not there, sir.
Okay. Cursor clear?
No, sir. I can't able to
Yes.
see.
Now?
Just you carry on explanation, sir. That's it.
This way. Red color. Are you
Yes,
able to
sir.
see
Yes, sir.
red color? So the red color
Yes.
portion is histone protein. This histone protein wrapped by chromatin, right? The blue color. The blue color wrapped by DNA. DNA histone together form as a nucleosome. Right? This model was proposed by Korenberg and Thomas. Korenberg and Thomas. The term nucleosome was coined by Howdett. Model proposed by Korenberg, term coined by Howdett. Each nucleosome consists a core of 8 histone molecules, and due to the 8, this is called as octamer. The histone molecules of the core named as H2A, H2B, H3, H4. There are 2 copies, and this rounded by chromatin. Just wait. Do not understand. Are you able to see? Hello?
Yes, sir. Yes, sir.
Are you able to see?
Yes, sir.
Right. How many rounds here?
4.
4, right. Here the 4, they are H2A, sorry, right, H2A, H2B, H3, H4. H2, sorry, A, B, 3, 4, right? 4. How many here? 4. But the same number arranged backside also. H2A, H2B, H3, H4. H2A same backside, H2B same backside, H3 backside, H4 backside. Total how many? 4 into 2,
8, sir.
8. So this is called octamer. And this octamer, sealed by double stranded DNA. Are you able to see this
Yes, sir.
DNA? Now, this is here sealed by another one, H1. Sealed by H1. Right? Is it clear first?
Yes, sir.
Red color eastern proteins. Octave 8. Okay? And it is rounded by blue color chromatins. Now this is called nucleosome. Like this, here this one stand, and somewhere here one, two. Just imagine backside also. 3, 4. Sorry, 4. Are you able to see the marker
Yes, sir.
cursor, right? Clear. So this is histone, red color histone, nucleosome, and again this chromatin thread again attached to the histone protein. Like this histone protein keep continuing the entire the chromosome, right? Okay. Hello.
Yes, sir. Yes, sir.
Right. So the DNA part between the two histone, this is one histone, this is another histone. Between the two histone, this place called as linker DNA. This is this called as linker DNA. This called as linker DNA or internucleosome DNA or spacer DNA. right? The same solid model, simple. The histone protein. These two closely arranged. If the two histone protein closely arranged, that should be in solid model. Solid model, fiber molded model, same. Is it clear? Any doubt in this?
No, sir.
Hello?
No, sir. No, sir.
Right. Any doubt today, class?
No, sir.
Right. Let's join to next class.
Okay, sir. In the next class, we are starting extranuclear nodes, sir.
Extra nuclear, better to read yourself.
Oh,
Nothing to explain there. And do you have any
okay.
WhatsApp group?
Yes, sir.
Can you add my number? Because I will send the cell division videos.
Videos. Okay, sir.
Better to avoid the extra time. We
Yes,
should
sir. Yes, sir.
we should focus on the remaining classes. Do you have my
Yes, sir.
number?
No, no, sir. I'm not having. You tell me, I will write it.
90
Yes, sir. 90
10
10
766
766
775.
775.
You add
From the
this
4th
number
of
in
Jan.
our group. Huh?
Yes, sir. From the 4th of Jan, we are having practicals also, sir.
Okay, no problem. You can join.
Okay, sir.
Okay.
Thank you, sir.
I will send cell division in group.
Okay, sir.
Okay.
In the next class, we will start directly with the block 3 because block 3 and block 4 is somewhat complicated, sir. Genetics and mutations.
Okay. Okay. No problem. If it is possible, I will explain in online or offline also. Okay.
Okay, sir. Okay, sir. Thank you,
Right.
sir.
Right.